Does it get really tall all of a sudden?
It's locally pretty steep. We're going to go up some tightly spaced contours and then summit fairly quickly.
Yeah, I'm just looking at Atalanta view. I'm like, ooh, we got some sheerness happening.
Yeah, it's not very tall.
Okay.
I mean, it's decently tall, but it's...
We got wall.
It's definitely wall.
Wall.
Wow.
For a little volcanic cone, it's pretty small. It's cute.
Exactly.
It's adorable.
Manageable size.
I like the weird bubbly texture
wall.
in the center.
Looking over the top of it already.
Yeah, it's not massive.
Oh.
That's another geology term I like. Massive.
That means not what you might think.
It's not. Yeah, no, I know, but like, it's not. This doesn't mean what you think it means. We're like massive massives.
Yeah.
What does it mean? Like, what's the?
Coming up.
Massif is a structural term.
Yeah.
And then there's massive in the geological sense, which just means not much in the way of features.
Oh.
Yeah.
Huh.
It's better than saying boring.
Yes.
Yeah, Levi, keep coming up. Because I had to come down some because this is kind of a risky thing when you do that. If you stay too close when you're right above it. Of course, we need to keep coming up because you're getting close to the wall now.
Oh.
There's a nice Faria.
Wow.
Are we still moving?
No, we're not moving.
Good.
Roger that.
All right. Yeah, come up faster. You got to keep that surface stretched out. What can happen is the tether can get a loop underneath it and then hang up on things on Herc. That's not good.
Oh, look, it's a little red shiny thing.
Ooh.
Right, went off the right of the screen.
Oh.
Another one of those sea cucumbers there.
Oh yeah. Ooh.
I think it's a ctenophore.
Ooh. Shiny.
Now these are predatory, correct?
Yeah, so they have those really long
Whoa.
filaments.
Oh wow.
Yeah, and you got the
Look at that.
Shiny
little cilia that are beat and look like little rainbows.
Oh.
Oh, and there it goes.
Oh no.
Okay. Carrying on. Is that another one down there?
There's a shrimp.
Yeah.
Ooh, those shrimp.
All right. I got to get going. No more
Alrighty.
sightseeing.
Sounds good.
The ctenophore was worth it. We haven't seen that before.
It was gorgeous. Ooh.
There's another cutthroat eel. We usually see a lot of cutthroat eels on the tops of seamounts.
Oh.
They're one of the top predators here.
Wow.
We got the cool striations coming back in,
Yes.
like the other nubbin.
Yeah, some striations, some joint sets. Kind of doing some quick math here. I think they're more or less on the same oriented the same way as we saw in the last nubbin.
Yeah.
Yeah.
Is nubbin an official
It is
term?
not.
It's our term. Can we look at this?
I think it needs to
I'm
be.
probably going to get in trouble with somebody for using it, but I will cross that bridge when I get to it.
Wait, I've been putting on the logs nugget.
Oh, there's a little shrimp on the side too.
So is this one of the same yellow corals
Yeah,
as
that
we
we
saw
saw on
earlier?
the other one.
Okay, so probably different from the yellow one that was collected on the 4 to 8 watch.
Yeah, I would assume that these would be different.
Okay.
Do we want to collect this?
Hang on, we've got both Fanny and Steve typing right now. So stand by. Steve asked, can we look on the plane? Fanny says it's not the same one that they collected.
She's interested.
There's interest in a possible collection here. So I think Steve is asking if we can rotate around and get a better look at the, so we're looking at it on edge and we want to look at it on the side, if possible. It's an interesting branching pattern.
Yeah. Turning around definitely gave a cooler picture there. It's so beautiful. Oh, is that a brittle star on it? Or
No, that's a snake star.
a snake star?
All right. It's all you now.
Okay.
Oh, wow.
All right, that's max zoom.
Good enough? Or
We have a request, if we can, to collect the whole colony.
Okay.
over collecting?
Do we have space up front to put that?
Up front? Yeah.
This is starboard
Okay.
There's a
firebox
sponge on
thing?
lambda.
We could probably stick it at starboard.
Yeah, we've got space in the starboard boxes too. I think C and D are still open.
C and D are open.
Can we zoom all the way out? I gotta set up better here. We're on this weird side slopey, not grand spot, not close. Give me a front faster, there you go. Okay, are we all stopped?
Yes.
What are we doing?
We're not moving.
And can you, Levi's, set up for the starboard side collection? Do your camera action. Don't switch the view, just get the cameras ready. I guess they are ready.
We are ready.
We zoom in. Ah, it's so snappy. I don't like it. Hang on. Force 2. That's better. Can we tilt down a little bit? still.
Ooh.
Oh.
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 Oh man.
Oh, that's a tricky one.
Coral cutters didn't cut.
No. I kind of messed that
So it's
up.
definitely not a brittle coral.
No, they're pretty strong.
Yeah.
Some of them are like, yeah, like nylon line, and then they're really hard to.
Yeah.
Did I get it?
I think you got it.
Yeah.
Looks like it.
Okay.
Nice.
All right, but we need to get this thruster off too. Oh, wait. Oh. Okay, can you switch to sample mode? What box?
C or
C or
D?
D.
C or D?
C or D?
Mm-hmm.
Is it blowing in the wind? Can you tell?
No, it's not blowing. Do you have more wrist rotate?
You want to flip it over?
Yeah, flip it over. Yeah, there you go. Oh no!
Oh.
Oh, it's that was a mess. This needs to get calibrated. It starts to spin on its own. See how it's doing that?
Yeah, that's
Oh.
not good.
That's bad.
Got too many. It didn't get in the box. It's like sitting on
No.
top of the lid.
It's what?
It's sitting on top of the lid.
It's on the lid?
Mm-hmm.
Oh, yeah, I can see it. It's in the, you can see it in the left camera.
Did that go somewhere?
I think it might have tilted in there. I can try bumping the box closed.
Yeah. This isn't a piece of it right there, is it? Can't see very good.
Yeah, I think it's in
is
there.
it starboard?
Okay.
Port starboard lights on? That would help.
They're on.
Ooh,
Doink.
Yeah, it's hard to see down there, but
yeah.
Yeah.
Well, I think it's in there.
okay. Yeah,
Okay.
we'll look for it topside.
Yeah.
That would have been better to have that light on.
Ooh, that was a tricky sample. Nice work with
Yes.
that.
So maybe in D? I think it went in D.
Sorry.
You did your best.
You're good.
Yeah.
No, some of these are tricky.
Sometimes it works, sometimes it doesn't.
Yeah.
Yeah. Like I thought all corals kind of were brittle like that. I didn't know they had internal structures that. like that.
Some of them can be pretty bendy, and this looked like a
Yeah.
bendy one.
Yeah, the black corals are notoriously challenging.
They are, yeah.
This one isn't black coral, but it has a similar kind of skeletal structure.
Oh.
And it's small, which is another layer of challenge.
Yeah.
Sometimes these corals are like way stronger than you anticipate.
Yeah.
Like that soft coral on the one dive in the monument.
Oh, yeah. Oh my gosh, that poor...
It
That-
haunts my dreams.
Me too. All right. So I think for just dive management stuff, the last 40 minutes of our watch, as we keep moving uphill, I'm on rock watch. I don't want to mess with starboard C and D though, because I don't want to accidentally flush out any coral pieces. But if we
Right.
get a small enough chunk of rock somewhere along the way, we might be able to put it on top of one of the big ones in starboard A.
Okay.
Starboard E or F, so in one of the big boxes, because I think I saw in E there was a larger rock.
Yeah.
So we can tell apart a big rock and a small rock. But I don't want to put it on top of anything in the front boxes.
Well, we can always go back to the video and look at the rocks. We do a good rotate. You could probably identify them decently.
Exactly, yeah.
Can we look at that little coral?
Zoom in.
Ooh.
What were we talking about?
Oh.
This?
That's the one. I think it's an arella.
Whoa.
That's almost more associate than coral.
Yeah, I know. Yeah, I was
It's
like.
got a lot of brittle stars on it.
Yeah.
Oh wow.
It's like, it looks like it has at least 4. Maybe more, 5 or 6.
That's so many.
Is it possible for the coral to get like smothered from the associates, like in a
yeah,
battle?
I think so.
Yeah. It's like that's so many.
It's the hug of death.
Yeah.
Depends on how long they stay there.
Yeah.
A lot of these ophiacanthids will jump off.
Oh.
I'm actually surprised none of them bailed when we landed. They do seem to be extra sensitive to the ROV.
Yeah.
Okay, we can move on.
Zero 20
030.
030.
Bridge nav.
Is that a little sponge over there?
You can see quite a bit of jointing on this bit here, forming that kind of stair step look.
Oh yeah, it's like super zigzaggy.
Yeah.
What does that stair step look mean usually?
That's probably related to cooling of the lava, but yeah, I don't have a good enough sense if it's aligned with the more regional stress field we've seen on this seamount.
Oh.
But yeah, definitely part of that, if not all of it, is related to cooling of the lava and how it contracts as it cools.
Wall here a little bit. Not big.
Yeah, a little bump.
Coming up.
The walls seem to be right where the contours are.
All the dead sponges. There might be live ones up here, possibly.
Maybe. But I've noticed this on a lot of the seamounts in the Pacific. Those really big Faria sponges that are dead and like hanging out on the bottom. And my personal theory was those were extremely abundant at one time in the past.
Yeah.
And then they all kind of died off probably around the same time, is my personal theory.
Yeah, because we don't see live large sponges.
Yeah, we see some of those, but like they're rare.
Yeah.
And it definitely don't account for the number of dead ones that we see.
Yeah. Yeah, I've been really curious about that. So I think we've seen this pattern in nearly every dive this expedition too.
Almost every seamount seems to have those massive Faria dead sponges everywhere. They're extremely common, the dead ones, but the live ones aren't super rare, so it's kind of crazy.
It is, yeah, and it makes you wonder, okay, what event is that tied to?
Right.
You know, when in time did that die-off happen, and when were they really abundant?
It's like they could have died off slowly, like over different time periods, but I don't know, that kind of seems really long time.
Yeah.
They don't really break down at all.
Yeah.
We definitely see them with like manganese crusting.
Yeah, I think we've seen a lot of those.
Oh.
So thousands of years or tens of thousands?
If you're seeing manganese crusts on them, you're getting into the hundreds of thousands to millions of years.
Wow.
Wow. Okay, they do stay around for a while.
Because the sponge is basically made out of rock.
Yeah.
These are
Pretty
silica,
much,
so yeah,
yeah.
actually.
We want to look at this or carry on?
Oh, we can look at it.
Cool.
See if it has the associate on it. I think it does. And then we also have a crinoid. So this is like the one that we collected earlier.
This is a coral or a sponge?
This is a sponge. And then on the sponge, there is a coral.
And those are the tiny little polyps
zoanthid
there, yeah.
Polyps, yeah.
Wow.
Kind of reminds me
like
of
little
octopus.
shrimps up here on the crinoid.
Oh, yeah. Oh, they're really small.
Yeah, look at
Little
that.
tiny shrimps.
Good place to hang out.
Oh, and I think there's, you see that little yellow bump on one of the branches?
The little node-looking thing there.
Yeah, I think that's a polychaete.
Okay.
A special polychaete that feeds on crinoids.
Whoa.
You answered my question before I had a chance to ask it.
That is so cool. Crinoids up close just look so ancient.
Well, they are.
Wow.
Right.
Yep.
Coming up.
I think there's some zoanthids on that dead sponge too.
There's a rock, but it's way too big.
Yeah, it's way too big.
Way too big.
Oh, there's like a little swimmer right at the top of the screen. See that?
Oh, zip.
Shiny.
Zip.
Oh,
Oh,
Yeah.
is it gone?
yeah.
Ah, too bad. That looked interesting.
It did. Yeah, we actually don't have sample bags down in the lab large enough for that kind of rock. Found that out with our 60-pounder.
Oh, jeez.
The texture of the rock here is really fun.
It's incredible.
Kind of looks like when you make focaccia bread and you crunch it together.
So what makes it have all these little pillows? The little.
So I
what's the
think
flow
we're
like?
I think we're seeing a lot of cooling joints again here. It's just we're seeing them in cross section instead of kind of along the sides of them. So these might be the part of like a hexagonal structure, sort of a columnar basalt.
Huh.
Yeah, so I don't know if this is the side of a dike or not. It might be. Wow. And if it is, which I'm reasonably confident it is, that would explain why we're going up a really steep wall here. We're just looking at the side of a dike that's been exposed. Yeah. Ooh, that was a good roll.
Yeah.
Oh my gosh.
Yeah, the weather is a little less ideal today than it was yesterday here, so we're getting a few wave sets here and there. And we just got a decent one that we felt pretty significantly up here in the van.
Yeah.
Yeah, the jet pump didn't like it either.
No, it didn't. Yeah, I could hear it working last night, off and on too.
The sea's pretty calm.
It's pretty calm until it decides to go roll.
Just a little squall rolling through.
Yeah.
All right. When do I hang out and wait for you?
Everything ship shape up at the bridge?
Yep.
Excellent.
Bridge says it's excellent.
Oh.
Just, you know, just wanted to roll.
Okay, good. That's reassuring.
Oh, wow. We have a pretty good question from somebody asking what kind of geologic or tectonic activity is going on down here? Or I guess based on what you've seen, like what are you kind of thinking?
That's a great question.
Yeah.
I think there's a little bit of both that's going on. We mapped this last night, and it's sort of an unusual feature that does have some fabric to it, if you will. There are a number of ridges on the side of the seamount that are broadly aligned with each other along a southwest to northeast direction, which tells you that there is at least a local stress field around this seamount, and it'll be interesting to see if we see that throughout the sanctuary as we continue to map in this area. There's also a seamount structure here. So it has some ridge-like features, it has some seamount-like features, and there are also a number of these, what we've been calling nubbins, here on the ship. A number of small volcanic cones, probably very short-lived, like single episode kind of eruptions, peppered across the summit, the flat summit of this seamount. That's a little bit unusual, and we're trying to figure out whether or not this might be a second stage of volcanism after the seamount was built, and whether or not it might have developed a carbonate cap, and if we have basalts that have erupted through that carbonate cap, if it's present. Because these little secondary cones might represent some sort of secondary phase of volcanism that could be potentially related to the evidence of some tectonic modification to this, the structure of the seamount, or they could be related to just a really late phase volcanic activity related to the original volcanic platform building stages. So there are a lot of questions that we have about structures like this, because you see these in a few other places in the ocean basins. They're not particularly common, so I don't think we understand them that well. I know I certainly have a lot to learn about these. So it's an excellent question.
Yeah, that's why we're here. What are we looking at?
This is a Calyptrophora wyveli, and there is a snake star in the center.
Ooh.
It's in the family Euryalidae. We're doing a little pilot training in the front row.
So yeah, we've seen a couple of these on this dive, so.
Yeah.
Yeah, they're really striking. I like the branching pattern on these.
Yeah, they're a pretty good one, and they're quite charismatic, I would say. You know, they're very unique. They've got a really cool look to them.
Yeah. They kind of remind me almost of like a weeping willow tree, sort of.
Yeah, yeah, it does kind of have that look to it.
I'm a big fan of that like grayish, purple, pinkish color.
Can I reset your DVL?
Pilots are moving us on.
Alrighty.
So we'll continue uphill and just see how far we get in the last 20 minutes or so, and then after that,
we'll go to lunch.
Yeah, after that it's lunchtime for us, and the next watch will take over probably for one little glimpse of bottom time, and then the blue water. And then we will recover after lunch. We hope to have the vehicles back on the ship at about 1 p.m. our time. This is a 14-hour dive, so we should be back up by 1 p.m. It's about an hour up.
Oops. I was muted. When are we going back in?
In the water?
Yeah.
Oh, I saw it on the board, but we didn't get a picture of it. I think we have to transit, right? Yeah, we have to transit,
time.
Probably tomorrow morning.
Yeah, because we're on the north end of the sanctuary right now, and we're going to transit down into the southern half, southern third, thereabouts. So there is a little bit of transit time there.
And these cucumbers seem really common here.
Yeah.
Yeah, the green ones.
I'm kind of tempted to grab one.
Slurp it.
We've got time.
I don't know.
I'm not
Did you
I
put
like-
a move in?
uh,
Did you?
I did put a move in, but you know, I'm just gonna put it out there to the science.
There is this is a smaller one.
As a smaller one.
That means it's slurpable.
Yes.
Yeah.
And
Very
we do have
slurpable.
a slurp jar open. I'm not going to say no.
Do Steve and Fanny have anything to say in the chat?
Chat's quiet right now.
Oh, okay.
That camera is really herky-jerky.
Yeah.
That's horrible.
To slurp or not to slurp?
Slurp. That is the question.
And I am not seeing any rubble around here, so I'm itchy for a rock, but I'm not sure we're going to get one before the end of the dive.
Let's slurp it. I think
Okay.
we can get one pretty
Yeah.
Okay, let's do it.
quick. You know, because we don't really know what it is, and we've seen a lot of them. So
I
they
think
are like very characteristic of this seamount. And we do like to collect things that are characteristic that we don't know.
And I think that is an excellent justification. So I'm right on board with you.
Slurp.
Slurp, slurp, slurp.
Slurps.
This one's going to be a very satisfying
Amber,
slurp.
can you open
I'm
up the
so
iris a little
excited.
bit?
Yep.
Thank you.
Told you it was going to be satisfying.
Oh, beautiful.
All right.
Nice.
Nice
Wow.
collection.
Hello, little
Well
friend.
done.
All right, cool. That
All right.
was totally worth it.
Nice and easy.
Yeah.
Just for that.
Yep. And that's very satisfying, especially after our last couple of attempts to slurp.
This is
Should we rotate
170?
the jar so it doesn't
170, yes.
Yeah,
Slurp
we can.
success.
Yeah, do we want to move it over to flush?
Anyways, can you rotate the jars to flush, which is the one with the red on it? So it's
I think that should be 5, correct?
looks like a counterclockwise rotation. The other counterclockwise rotation. Just
nope,
keep
I was
Just keep going. Once you're committed, just keep going.
Never mind me, I can't count.
There.
There you
Oh, yay.
Charge that.
Yeah, that should be 170 there. Excellent.
Slurp, slurp, slurp.
What jar number is that?
Jar 7.
7.
Oh, right.
All right, moving on.
go. Cool.
Yay.
That camera is really,
Yeah,
Isn't it?
I don't watch that one. That one
I know, but
kind
it's
of
like
gives
worse
you
than
motion
normal.
sickness.
Yeah, it's worse than normal.
Yeah, isn't it?
Yeah, by a
You want
lot.
to cycle it, or what do you want
You
to do?
cycled it once and it didn't seem to do anything.
It didn't do anything?
No.
Mm.
You try
Okay.
again. I'm open to that.
Do you want to try it again?
Yeah.
Levi, do that.
Yep. Lower cam.
Lower cam.
Is that the same as cinema cam? Because it looks okay back here.
No. No,
Okay.
cinema cam is
Different.
mid.
Lower cam off, I think.
Leave it for a while. And then... All right, turn back on.
Lower cam on.
Oh, a tiny fish.
Little fish.
I think that might have been a mactophid
Ooh.
or a lanternfish.
Aren't those? Oh, we're pretty shallow, aren't we?
Yeah, we're shallow, really shallow. We're at the top of the seamount, so we're kind of getting that
Oh.
mid-water
Can you zoom in?
interaction.
Oh.
I only based that ID based on how it was swimming. It was kind of darting.
Yeah.
Yeah. Very dark colored crinoid. Looks like maybe a tube worm skeleton in the upper part of the screen.
Tube worm?
Or anemone or something. Right there.
I think it's just a base of a dead sponge.
Oh, okay, another sponge base.
All right. Carrying on.
Cool.
Sounds good.
Yeah.
I think I'm
Is
not
there rubble
squared
in
up
this
here.
thing? Nope, that's
Pretty
all
square.
cemented.
Just being picky.
No, okay. Oh, look how square these are.
They are
Yes.
very jointed.
Ooh. It's Minecraft.
So those have your
We love
columnar
Minecraft.
basalt look, huh?
No.
I think
No?
this isn't columnar. This is, let's see, 29 degrees. This is aligned with the regional stress field. This is one very stressed out seamount.
Oh man, so stressed.
Rubble.
Poor thing.
Can we look in here for a moment?
Oh, see
In
if there's
the
like a
rubble
little
pile?
rock.
Yeah.
Ooh. And are all these corals and sponges, or just?
Give me
Yes.
some front basket.
Okay, front basket coming in.
Yeah, we might be able to pull our last bottom Niskin
good.
I
here.
think that's a good idea.
Yeah, so grab a rock, grab a Niskin, and then kind of get things wrapped up and ready to go blue water for end of watch.
Yes. Sounds
Okay,
good.
sounds like a great little work plan to tie this one off.
Yeah.
These aren't very angulator.
That's okay.
Not being picky, huh?
I would be interested in anything that's loose around here. That one is interesting.
All right.
That might be our best
We'll
bet.
poke.
We will poke.
Sounds very rectangle shaped.
Not going to make any promises, but we're going to give it a try. And there are a couple things we can do around here. Just
Okay.
tie a bow on this dive.
Nope.
Ah. Yeah.
No.
Nope, we're moving the vehicle. It's not going to move the rock.
That rock is stuck.
Yeah, they do that. That's why I wasn't making any promises.
Dang.
Yeah, we should have 5.
Everything's in place.
You can write your
Okay.
name on it though.
Robert was here. Okay. Well, we do see a higher population of things in this area, so I think Backrow is interested in
Niskin?
pulling one of the Niskins
Niskin.
here.
Yeah.
Okay.
Hey.
What's that thing?
Ceranthid. It's a tube anemone.
Can you give me some more iris?
Yep.
And we were going for number 4, right?
4,
Yes,
yes.
correct.
Orange
Cool.
one.
So we got 1, 2, 3, 4.
See, that was also a nice thing about the balls. They had the numbers on them.
Mm-hmm. Printed on, too, if I remember
They
correctly.
were printed on, yeah. Very satisfying.
Yes.
I'm not going to argue the balls and the rings anymore. Are you watching?
Watching.
Watching.
Yep.
Yep.
Done.
Yay.
Nice.
Success.
All right, onward.
Yes. All right, so we're coming up on the last 10 minutes of watch, and the next watch is starting to work their way in slowly.
The fuzzy
so.
one. That's new.
Fuzzy
Yeah.
one.
Yeah, the fuzzy one.
Ooh, fuzzy wuzzy.
Is that a chrysogorgia?
Whoa.
No, that's something different.
It's a black coral.
Ooh.
That is the fuzziest black coral I think I have ever seen.
It could be a lilipathes.
It's so fluffy.
There might be some interest in science sampling this. I'm
Okay,
not
do
sure.
we want to do a quick sample?
The black corals are really unknown. I don't know what this one is.
Right.
Lilipathes is my best guess.
Okay.
I need reference here.
Oh, and I think there's a madrepora too.
I need
Look at these.
I'm sorry.
Grab all of them.
And I'm over here looking at the samples, and we're
kinda
nearly
full
full
up.
up. So I think
What
this
is the
might
plan
Coral, we
here?
could do lambda.
we could put it in
well,
lambda, yeah.
I feel like
Forward by a box.
Steve or Fanny should decide if they want the black coral or
Oh,
the
look.
or the. There he
Video
is.
watch
He's
change.
right here. He can decide.
I'm a big fan of the fluffies.
We don't know.
We don't know. Maybe in CND.
Alrighty, y'all, stay tuned. 8 and 12 is currently signing off and doing a watch change. Again, if y'all have any questions, tune into nautiluslive.org, and the next team will be really happy to answer. Thank y'all for watching.
Which way are we going, and which way is Atalanta pointing? Let's take a look. We're going uphill, right? So we're going to the north.
We should be going uphill. Let's
Northeast?
see where we are.
Atalanta's looking...
Yeah, we're almost at the top of this.
Okay. All right, I got it.
We're just climbing the mountain.
Okay.
All right, so since we don't have a lot of time left, I'm actually more interested in over that.
Ooh, okay.
Yeah.
Understood. Head that way.
Sorry, what do you say? More interested in...
See that coral in the upper right?
Awesome.
We're gonna move the ROV that way.
There you go.
I know we're not fully changed over in the back row here, but I'm trying to make every moment count.
Yeah, exactly.
Thank you for chiming in.
This is a 8 to 12 watch takeover by the 12 to 4 watch lead. But it's for a good purpose, I promise.
Coming in from the Natural Museum of Natural History, Steve Ostovich, with you live. Back to you, Steve.
All right. So we're going to take a look at this big colonial scleractinian here. And I am pretty sure this is going to be a collection, but it's a bunch of fragile rubble, so we're going to want just like a big chunk of it. You know what I mean?
Roger. Chunk.
Yeah. Yeah, I think forward bio box and the left-hand side, lambda. But can we get some good imagery first?
Yeah.
Yeah, absolutely. You good to go, Levi? I zoom?
1 second. Let me
Cool.
center it up. Auto XY. Heading 423.
I want to see mostly if there's any associated animals in this, because this is kind of a big reefy clump of, I don't want, it's maybe a stretch to say reefy, but it's one of the species that is known to make deep water stony coral reefs in the Central Pacific. Yeah.
Go up closer for you.
Yeah, it's a stony coral, a scleractinian, colonial, in genus Madrepora. And this genus is actually undergoing revision right now, with a few new species being described recently, and more on the way. So it is important for taxonomic work and may represent new species or new records for the area.
Very cool.
All right, videos come in.
Copy.
For those of you watching, we are going through our shift change. Our 8 to 12 just ended their shift, and our 12 to 4 is hopping on. Be right with you.
That is full.
Feel like I'm drifting away on you.
Yeah. Can we look more on the left side, I think? Okay. Let me get a capture.
Copy, coming out.
If you need me to take the capture, I need a half-second login.
Oh,
but
sorry. I thought by capture. My brain's still waking up. I heard sample in my brain.
There
Okay.
are a few squat lobsters on here. So here and those at Brittle Star, but maybe here there's another. So there are associates, and there's some barnacles associated here, and maybe anemones too. So these are things we wanted to document. They may or may not come with the sample, but I think it's important just to get a big clump of it, and you're probably going to use a soft grip force to, otherwise it's going to be pretty trashed. But there's not much more we can do to.
Are they all attached to this
And
like brown stalk in the middle?
Yeah, assume it's all attached. So.
Yeah.
Yeah.
Are the white pieces dead?
White pieces like this, yeah, that piece right there has no tissue on it, but if it's brown, so these are with tissue, and you can see the polyps and the tentacles.
Gotcha.
And then, sorry,
Yep.
What are we looking at?
This is a Madrepora.
Madrepora, and then you said brittle stars, squat lobsters, and barnacles in association?
Yeah.
Thank you very much.
All right, coming
All
out.
right, I'm good to go whenever.
Data, what's the next sample number?
This will be 172.
Thank you.
So I think make it easy for yourself. If it's easier to grab, a chunk from that side, I think that might be less invasive. Maybe just go for that.
Too far away. But what do you think?
Whichever is easiest for you, that's fine. Just know it's going to be brittle and it's going to shatter as soon as you touch it.
Which box is it going in?
Probably forward bio box would make the most sense because we have a sponge in there.
Yeah, we're going to go
the
there.
starboard bio box, they didn't know which one a coral landed in. So either C or D has a coral in it.
Yeah, I think we're going to go forward lambda with the sponge, so forward left.
Okay, thank you,
Is it
Steve.
Yule going by?
Make it easy on y'all. Yeah. Absolutely. So this is also commonly known as zigzag coral.
Ah, I can see why.
Yeah.
That definitely has that characteristic design. It's zigzag.
Yeah, if that's where you're going, that looks fine to me.
Sorry, I wasn't on SPL. Yeah, okay.
Yep.
I'm going to do that.
Yeah, very interesting morphology. Is this the first one of these we've seen, Steve?
Yeah, we're kind of in the peak depth range now.
Okay.
Oh, that did not grab
wow,
much.
super brittle.
Here, I'll try again.
I wonder, wow, that is really crumbly.
That is like crumbled
I
before
warned
I
you.
even get the job. Here.
Yeah, you're going to just kind of have to get a grip of it.
All right, there we go.
Right.
Got some in there.
I'll zoom out on me.
Looks like we got some associated.
The nice part
too.
about these scleractinians is that whatever their orientation, they will cement back down to the seafloor and continue to grow up. So if you think about how a coral reef grows, corals just settle upon each other, and these are very much, not only are they evolutionarily the same group, but they're also,
Left side, yeah.
they behave the same way.
Okay, yeah.
They're
Yeah,
just not as big.
Right.
Yeah.
Yeah, it looks more like a shallow water coral, at least to me, than some of the other species we've been seeing.
I'm going to let it go there.
Yeah. That's a nice grab. Yeah, happy with that.
Okay.
And if we can zoom back in on the rubble, I just want to take a look at a couple things.
Yep, just
Good
gonna let them
job,
close up.
pilots.
Rubble.
That is a crumbly coral.
Yep.
I'm going to extend that camera back out.
Yeah.
Thank
We're
you.
going to go for the rubble.
All right, coming back in.
Yeah.
And I want to kind of zoom in this area
Yeah.
as tight as you can get it.
Okay.
Okay. This is kind
There's
of,
something in there.
so you see this squat lobster? You
Yeah.
can suction that. Do we have a bucket available? There's also a squat lobster here.
It's feasting.
Data, maybe you can confirm we don't have a
We do
bucket.
not have a bucket available. There's the flush jar, but.
Yeah, we could do the flush.
Let's do the flush. This one's kind of important.
Okay.
And then,
Now to get your
are we
cluster.
counting
coming
this as
wide.
part of the same sample, or is it a different one?
The nozzle looks like the green portion has slid even
Yeah.
further forward.
I would call them the same sample.
All
They're
right.
just
Can you
one's
take
an
it over just to
associate. The
take
problem
a
is you just
look?
can't collect them together, but
I mean, about
it would be better to have them linked in the data
yeah,
record.
Probably will have
And
to.
it won't be the first time we've had a sample split between.
Yeah, it happens all the time, yeah.
Otherwise we might pull stuff off.
So this is probably going to be a Munidopsis. And what's interesting about Munidopsis that are associated with stony corals is that they're likely different species that are associated with stony corals and which are associated with either free living in the environment or on octocorals.
Oh.
And actually one of the new species that was described from this genus from the area around Jarvis Island, the cruise was in 2019, but the specimen was described this past year. It was associated with stony coral rubble.
Oh, wow.
Yep.
Zoom in video,
So this may help us learn a little more about that.
Yeah,
Absolutely.
Very cool.
Coming in now.
and it's really important, you know, one of the features that makes coral reefs important
You ready for inhale?
is that
Oh,
they,
yes. Relay.
Oh, sorry, I didn't get
Camera.
anything set up.
Bucket. Bucket. I'm on flush.
Yeah.
Here we go. So this might just hydraulics 81%. I'm gonna reduce it just a little bit.
Yeah, otherwise we might suck everything up.
Oh,
He's like,
I know, he's like, get away.
Get out.
Alright, you got suction on?
I should be on.
Alright. Do we have any?
I don't have indication, if I'm being honest.
I don't see. Oh, I'm trying not to. Here. I'm gonna. I just don't want to hurt the rest of the coral.
It's a rough angle.
I don't feel like we have suction.
Okay. All right, killing
Maybe
it.
power cycle that.
Oh, yeah. And back on. Back on.
Alright.
Okay, I think I saw a speck fly by,
Okay.
but it's hard to see.
Yeah, suction's on.
You see it?
Order.
You see flow? Okay,
Yeah.
I'm ready on the kill button. It's not moving.
No.
Oh, no, I did see something. Oh,
Oh, is that in there? Something's, oh no,
Okay.
it's
Coral,
rubble.
coral. I'm just gonna kill it. I'm gonna go with my gut.
Yeah, I think it's in there.
Oh, it's in there.
It's in there. I
Nice.
see
Index
it.
off, index off,
Nice.
index off, index off.
Good job,
And it
guys.
sunk down to the bottom. Perfect.
Yep, there it is.
Okay,
Well, this
so
is jar 1 now, so I've indexed away from it.
All right,
so
coming
let me go
wide.
back to, what's in? It's hard to know which jar I'm supposed to be on now.
So
'cause I don't
just
want-
for your information, Steve.
Yep.
There is a chance that because you have a sample in there that's a moving creature, there's a chance it escapes from the jar as we come up.
Yeah, maybe we should just leave it on the sea cucumber jar
Okay.
I
for
left it on
recovery,
the-
because that's not going to
it's
escape.
cup
That's
coral. Cup coral won't escape, right?
I don't know.
It won't. It could always get
No, I'm kidding.
flushed.
That won't
Coral
escape.
should be good.
Yeah, I would leave it on that or sea
Yeah.
sea cucumber. Yeah.
Okay.
I saw the squat lobster go in the flush jar, but when I indexed off of it, I didn't see it.
I
hope it's
did
in
see it. It's in there.
I hope it's in there.
100%.
All right.
I did see it. Yeah. That was a great collection. All right. How
Awesome.
much bottom time do we have left?
Actually, can we zoom on that slurp? I just want to make sure that green thing's not about to come off.
I think the hose clamps look pretty tight, so I think it just slid down the length of the hose.
I think it's off.
One of the clamps is loose.
It's
Yeah,
off.
definitely.
Well, so one of the purple clamps is totally off. That's the loose one.
Oh,
but there's
there's
2 on the green.
There's a cup coral
I
there
poke
too.
Yeah.
Are you gonna try
This
to?
is,
I see PVC after the green.
Yeah.
Stony coral
that?
little peak nubbin
You
here.
do?
Yeah.
Yeah,
Oh,
there's a
zoom in,
little
zoom
lip
in,
of,
zoom
yep,
in.
yeah, I see what he's talking
green,
about.
The green is totally on the
Oh,
PVC.
See?
it's an
Yep.
optical illusion.
So
I see.
it's
Okay.
not. Yeah.
Okay, we're good.
We're good. The green piece should hold on.
So
Okay.
that one
Yeah.
hose clamp is like kind of cocked a bit. Yeah.
Yeah.
All right, coming wide.
All right. Thanks, guys.
I'm going to give a quick shout out to my family in Bridgewater, Massachusetts, the Bairds. They're at Auntie Trish's Sunday night.
Aww. If we're giving shout outs, happy birthday, Jack.
Oh. Shout out to my cat.
Right.
What's your cat's name?
Monkey. I call her Binky.
Monkey. That's so cute.
Shout out to Binky.
Gabby, if you want, I just switch the camera to you. You can give it a wave. You're right in the corner.
Oh, sure. Sunday night. It's time to go to bed, kids.
How much bottom time we got left here? Who's keeping me honest on the ascent time?
Do not know. Not
All
me.
right. Which way have we been moving?
We have been moving it's kind of like northeast. That's 030.
30?
Yep.
Okay, so to your port, co-pilot, looks like. So
Yeah,
if you want to turn port a little bit, looks like this is just a local high that I'm sitting on.
Yeah.
So if you turn port, I'll go with you, and then we'll go another move that way, maybe?
Yeah, sorry, what heading? We're
030.
okay.
You can just climb up.
Three.
And
Looks
how
like
much
we're
time
on
did we say we have?
I think recovery was 1, and it takes us 40 minutes to get up.
Copy.
So what does that put us?
10 minutes?
Yeah. Okay.
All right. So let's poke around the top of this nubbin and see what we see.
Ooh, is that a big crab?
It's, I think it's a homalid. It's a larger individual, what we were seeing last night with the
do you want,
anemone on the back.
Yeah.
bubble on center cam?
Yeah, you can leave it. It's fine. You can just leave it. Yeah. Thank you.
For those of you just joining us, we are
Ooh,
finishing
that?
the last little bit of our dive on a ridge off of Bocock Atoll in the National Marine Sanctuary of Bikar and Bocock in the Republic of the Marshall Islands. It was actually a recently established marine sanctuary earlier in 2025, and it is the first one for the Marshall Islands.
Crinoid.
This is why you always try to reach the top, because the good stuff's always on the top.
Yeah.
You're always surprised by what's on the top.
Y'all, the 12 to 4 saw an anglerfish.
Yeah, that's
So cool.
4 to 8.
Yeah.
Weird. Currently,
Aren't we the
around-
12 to 4?
Yeah, we're the 12 to 4.
Whatever. 8 to 12,
Still waking
whatever
up.
you call
Still
them.
waking up.
Still waking up.
We're currently at 951 meters below the
Tilt me
surface,
down,
and we
I'll land
started
here.
just over 1,300 meters below the surface. about 10 hours ago.
All right, good for zoom.
Yeah, go for zoom.
Crinoid?
That looks
On
like
a sponge?
crinoid on a Furrier sponge, yeah.
How many crinoids?
A couple crinoids.
Yeah.
3, right?
3. Yep. These are echinoderms, similar to sea stars and sea urchins. But these are
palm tree.
Yeah. These are primarily planktivorous. They're feeding in the fast-moving current.
Oh, sorry.
And their mouth is down, way down in the center of that disc, in the middle of them.
Okay. Where it kind of clumps together?
Yeah, there's a bunch of, usually small tube feet will help move particles towards the mouth.
Widen out just a little bit.
They're actually really cool to watch if you have the patience.
And this would probably be one of those free swimming ones?
Yeah, this is a Acanella,
Okay.
the group that the free swimming
Yeah.
crinoids belong to. They don't really like to swim, but they will if they are not in an optimal spot.
Gotcha.
But these pretty much have the optimal flow conditions.
Yeah, up on the top.
Top of a mountain, top of a sponge. Yep.
Getting all that good
All right,
current.
Cool. All right,
Looks
coming out.
We currently have people watching from all around the world, from the US, Canada, United Kingdom, Germany, Australia, New Zealand, Guam, and Austria.
Hello, world.
Hello, world. Stay classy, world. Stay classy, planet Earth.
This is interesting that we're going up the terrain in this heading.
So I think it's like a local bump. And
Gotcha.
then it goes to the global. Yeah.
So we're not going to little...
The local.
Little
Valley.
saddle.
Yeah. Little saddle action.
But there are, yeah, it's, I mean, have they taken a Niskin recently? I'm not sure.
We took one right, they took one right before we got on shift.
Okay, so.
10 minutes before. And then
Right,
there's
that's
2
area.
left.
Yep.
Is there a more scientific way to describe the benthos than pillow basalt that's kind of rectangular?
Hard.
I'll jot that down. Thank you.
Similar to rock.
I'm really stretching into my vocabulary there.
Another crinoid on the Furrier sponge, right?
Yeah.
I'm slowly learning.
Yeah, and then all of these other primnoids around them are the same, similar, no, it's the same species we've been seeing, called Calyptrophora wyveli.
Wyveli.
Did they drop any plates? Looks like maybe they dropped one.
Again, for those of you joining us, just a little bit about our objectives for this dive. So we are not only looking for biological specimens to help us learn a little bit more about possible range extensions or new species, but we're also looking for new geographic records and geological samples to tell us a little bit more about the geophysics of Earth and a little bit more about the history of life on Earth. So we'll take those samples up to the lab for further analysis, both our geologic and our biological samples.
In the water?
Video, snapfish.
Fish, snapfish, fish.
Oreo.
Whoa.
Did I promise it or did I promise it?
Is that an Oreo?
You've delivered.
I have delivered.
Oh
You
my
an Oreo
gosh.
somated
Steve,
So
you knew. Did you talk to that one before?
on the top. No, actually, is it? No, it's not.
Is this
Aww.
a hatchet?
It's hatchetfish.
Whoa.
Yeah. It has the same type of deep body, but no, that's a hatchetfish.
Hatchet, like the
Yeah.
mini
There you
axe.
go.
Yep.
Please.
Oh.
Swim away, swim away. I scared him.
I was so looking forward to having the Oreo fish too, but
Hey, we've still
alas.
got 5 minutes.
The funny thing is that they're often associated with those madrepora corals. So we're in the right depth range, we got the right habitat, we just don't have the fish.
Fingers crossed.
This lava flow is really cool.
It is.
It's pillow lava.
Another objective that we're looking for in this dive, as with some of our other dives, is collecting eDNA or environmental DNA samples. So we collect water using two different techniques, our Niskin bottle and our large volume multipuffer, which we will analyze in the lab, and it'll tell us a little bit more about what organisms live down here and kind of give us that big picture.
I'm just trying to stay looking at rocks in the last 5 minutes, Steve.
Yeah.
Because it's just sand up ahead, so.
Yeah, no, this is good. There's stuff to look at here.
Yeah.
A lot of old
Are those
bases of things.
Oh, a sea star.
So
right?
there's pretty good settlement and recruitment here. Things have been living here a long time.
Oh, tilt me down. I'm going to go for sea star.
Yeah.
We're looking for a sea star.
I don't see it.
Oh, yeah, there you
Oh,
go.
there it is.
There it is. Ooh. Oh, yeah.
You're not extended. There you
Oh,
go.
thank you.
Okay.
Oh, bonus.
Good for zoom.
Yeah, you can zoom. I'll try to get you lower here.
I think this is the same one we saw a bit earlier, too. Oh, yeah. Actually, this one looks very similar to Henricia. But, all right, that is full.
Roger, full zoom.
Very nice.
Yeah. Does it have like little pores on the top? Or are those just pigments?
They could be nubs of spines or, you know, small ornamentations on the back of it.
Okay.
I think this one is in the genus Henricia. All right, coming out.
Sweet.
Yeah.
Ooh, it's a matrix.
It's likely the video cable on that monitor, like HDMI or DVI. Because this monitor was fine.
Yeah. Yeah, it was. But if you see a black cat go across, then tell me.
You might be in the matrix.
Yeah.
Yeah.
I love black cats and black dogs.
It's your last 2 minutes, Steve, and then I gotta pack
Yeah.
up.
What's up to the right? Let's go to the top.
I think that's coral we sampled.
Yeah. Can we?
Like I think it might be the same colony.
Yeah, that's
like,
same. Yeah.
Like I circled back.
Have we gone
Oh.
up that way yet?
Yeah. I might as well take an extra look here.
Oh, look at all this.
Oh yeah, this is pretty much the top. Actually, there's another stony coral colony there.
Got it.
Yeah.
Oh yeah, there's one on the back side of that rock over there. See that on the left?
Yep. Yep.
Lots of stuff going on up here.
You can tell by the flow of the marine snow, kind of from your
the debris, kind of maybe.
Hazard to guess, but yeah. Can we
snap it?
Snap it? Yeah.
Yeah.
Copy. See if anyone else is home.
This isn't the one. Is this the one we crumbled up?
Knock,
No,
knock.
this is
Or is this a different one?
untouched.
Untouched. Okay.
Yeah, it doesn't have all the little pieces under it.
Yeah. So these, you can see there is another squat lobster in there,
Someone's in
right
there.
there.
Yeah.
And these kind of they find a really nice spot in the seafloor, and they just will continue to build upon themselves over time, and they will eventually continue to shed rubble downslope.
Oh, okay.
But these are, I mean, there is not a single drop of rubble on the way up, so these are not growing as much as some of the others we've seen on different islands and atolls at this depth. But this is the depth where you would typically find deep water reef development.
Okay.
It's just they're pretty small here still.
All right.
All right,
Don't even
one.
want to get into that.
Yeah.
Next video. I think that's end of dive, right? We're gonna start coming up. All right.
All right. Well, that was very eventful. I think everyone was really happy with the dive track and collections. We filled up the vehicle.
Lots of really interesting things on this dive.
Saw some probably 30 species of corals, 20, 30 species.
To be honest, I don't know.
Lots
other
more
marine invertebrates.
Lots more vertebrates as well.
Maybe we'll get
Fishes, the halosaur,
eel.
Halosaur aggregation was really interesting.
Yeah.
So
batfish, tripod
you want to turn
fish,
starboard a little bit?
cuskill. All sorts of good things on this dive.
This is such a good final view.
Yeah.
Yeah, this is why we always try to aim for the pointy, hard bottom stuff on the bathymetry, because this is prime coral habitat.
Yeah.
Yeah.
Amazing.
Coral and sponge, really. Sponges do well here too.
I'm gonna punch it out of depth so I don't float up too much.
Goodbye.
Pushing out.
Would we say we're heading off bottom?
Yeah.
Thank you.
You're off bottom.
Look at that Atalanta shot. That's a really interesting topography.
Yeah, it's really cool.
Nice part about finishing a dive shallow is the short ascents.
Yeah.
We'll be there in no time.
Not as much blue water.
And I think let me check about what we want to do for the last Niskins. I'm going to look back at the dive plan here to see what makes sense.
Auto heading.
Data, what do you have for off bottom time?
I have it in C log, but 21:26.
Thank you.
I think we also have
Wait, sorry, 0021, my bad. Continue, Matt.
Yeah, no worries. I think one of our previous SCFs, Shannon Celine, is watching from her classroom right now in Guam. So
Awesome.
hello, hello.
You can probably remove your auto heading. I think I should be tugging on you by now, right? Yeah, totally looks straight.
Yeah, shout out to Miss Celine in her classroom watching us right now.
All right, so we got 2 Niskin bottles left, right?
Yep. 5 and 6.
And away we go, heading up.
All right, so we'll have an on-deck time of 1300.
The oxygen sensor's working as expected this dive? I'm just wondering whether I should trust the data.
Every dive thus far, I've seen it as sensor wasn't working, but I don't know about this one. It looks like there is data.
I have cameras right here. No, we can turn it back on. Oh, thanks. You can start hauling in. I'll start pushing up here. All right, I'm pushing up. I'm pushing out. Tether's tight. About to 14.
Yeah, let's just go with 50 meters.
Yes,
We'll,
50,
we'll
copy.
pop both at 50 meters.
Sounds good.
As we've been doing.
Do you
I
wanna drop
think
a
we've
weight?
been calling them different samples
Can you get a second?
too.
Yeah,
Yeah,
I'm gonna
okay.
drop one of the weights.
because we only use one bag of each anyhow.
Is that what we tend to do if we don't use all the Niskins?
Yeah.
No, it's also useful for characterizing kind of the upper surface mix layer.
Okay.
So it is useful data. We just want to be consistent, see if there's
Right,
any
yeah.
spatial differences.
Awesome.
Okay.
The
Right,
science
retract
never
this
ends
bad
on
boy.
the Nautilus.
Ooh. Yeah, toss one of those. Copy.
I think that's what's supposed to happen, right? It's like a redundancy. Yeah, teamwork.
This is our gift.
Our gift to King Neptune.
Be free.
Nice. Should help our... Yeah, I agree.
And so do those removing the weight, does that just allow us to descend a little bit more stably? Or?
Yeah, so anytime we pick up rocks, I think Danny picked up a big rock, that's our ballast, so that helps us get up. Otherwise we go up too slow.
Gotcha. How fast does Hercules ascend?
Let's see, right now we're doing like 16 or 17 meters per minute, but we should get up to 25.
Okay.
Let's see what we're doing. 18 right now is what we're measuring.
Nice.
We're usually limited by the winch speed, which is 30 meters per minute.
Gotcha,
To safely
okay.
operate,
Yep, so again, those of you joining us, we are just finishing up our dive on a ridge off of Bokak Atoll in the National Marine Sanctuary of Baikar and Bokak in the Republic of the Marshall Islands. We are about 900 meters below the surface and hoping to be at surface in just a little over 30 minutes. I'm still holding out for Oreo.
Yeah, not in the water column.
Do you wanna,
Probably not.
are you comfortable starting to track a line nav? See how I'm drifting southbound? Are you comfortable having the ship come ahead and drag us out? Either, yeah, that's fine. Whatever recovery direction is fine. Just have them start coming ahead. Thanks. I'm gonna reduce my
As we're heading up, I guess we could go ahead and, for those of you that are just joining our dive or maybe joined since our last watch and want to know a little bit about who you're listening to, we could go through a quick round of introductions. My name is Matt Holden. I am a Science Communication Fellow for NA174. When I am not at sea, I am a high school science teacher in Fayetteville, Arkansas. Steve, would you give us a short introduction?
Sure.
Sweet, thanks.
My name is Steve Oskovich. I'm the watch lead for the 12-4 and one of the biology science co-leads on this expedition. I am really interested in researching marine biodiversity and specifically focusing on the molecular genetics and relationships among species of deep water corals. When I'm not on Nautilus, I work at the Smithsonian Institution's National Museum of Natural History in Washington, where I work on genetic sequencing and environmental omics of marine animals, largely deep sea.
Very cool. So a lot of your work can tell us a little bit more about new species and range extensions with all the genetics and things like that?
Yep.
Awesome.
That's the plan.
Very cool. Mariana, would you mind telling us a little bit about yourself?
Yahweh, I'm Mariana Poznansky, and I'm on the Science 2 position. This is my first cruise on board UV Nautilus, and when I'm not at sea, I'm an intern at MIMRA, and yeah.
Awesome. So excited to have you on board. Leela, would you mind telling us about yourself?
Yeah. Hi, I'm Leela Madhava. I am the data logger for this shift, which means I'm keeping track of important events and samples as they happen. And when I'm not at sea, I'm a student at University of Washington, where I'm going into my senior year, double majoring in biology and marine biology.
Super exciting. Do you have any plans after your senior year?
It's the million-dollar question.
Right.
I'm hoping to hang around Seattle for a year or so and see what job pops up. I'd love to do restoration work.
Oh, cool.
If it's available.
Nice.
Yeah, just
Very
whatever
cool.
I can get, pretty much. I'd love to come back to sea also.
Yeah, totally. Same here.
Yeah,
I'd
the
love
trap
to get a
another
chance to come back and work
You
aboard
think-
EV Nautilus again one day.
Yeah.
I mean, I'm only doing 18. At this point, I'm going to be late.
Let's see. Dan, would you mind introducing yourself?
Show me my porch again.
Yeah, absolutely. Name's Dan Krylov. I'm the video
camera is
engineer
usually
for this
seen.
watch, and when I'm not here, I'm freelancing
Those
for all
big
kinds
zip ties are
of
partially
live
cut, so it
production
should be
events. And
easier.
right now, I
I was
just
supposed to use the
snagged
little
the
zip ties.
sweetest freelance gig there is. So
Awesome. Yeah. Super great gig out here. Glad to have you, Dan.
Nice. Let's go.
Get out of here.
Come on, get.
Kicking it into overdrive today, huh?
Well, we're a bit heavy.
It's already, I don't
All
want to be
them
too
rocks.
All your rocks.
We do have
Val,
several
we got your rocks. We got them. It's rock o'clock coming up. Get ready.
Rock Express.
Rock lobster.
Oh, that was my first freestyle this trip.
Now, how do we get all those rocks back?
Nice, we gained.
back home.
Package them?
I could put them in big crates.
Send 'em out?
We
Freight
do
shipment,
have
right?
5 rocks, by the
wow.
way, which could explain why we're not moving so fast.
2 meters.
Steve, do you take all these rocks home and then you tell the TSA agent it's your rock collection? And they don't, is that how that
I
goes?
do not.
Like, what
I
is
do
in your
not.
bag? Glad you asked. These
Pick
are
up
rocks.
their bags,
Yeah.
like,
I think Val would
Some
actually
weird
love
rocks.
that if
And then we see
the
them.
TSA asked her.
Yeah.
What'd you pack? Whole bunch
And
of
then
rocks in here?
You see them in the glass
so
case
heavy.
of like TSA like confiscated items. Like, yeah, these are rocks at the bottom of the ocean. You're not supposed to have big rocks.
Yeah, I bet the
And
shipping
then you
cost
put it
on
on
that
loan
is
from the National
wild.
Museum or something. Like,
Yeah. That'd be even more sketchy if they're nodules.
Going a little faster now, aren't I? I'm making 20 now.
Nice.
I
ROV
really
pilots,
like this.
would
The
you
intro
mind
is
introducing
really good.
yourself?
Sure. Sitting ROV pilot flying Hercules is Levi Unima, and I am from Newport, Rhode Island.
Awesome.
Sitting co-pilot right now is Ryder Wolf, and when I'm not here, I sometimes do small vessel ops around Vancouver Island where I live, and I like my cat, so I spend a lot
Love
of
it.
time with my cat.
Love it. I love my cat too. And you also like long walks on the beach, if I'm not mistaken.
That's true. The war
I think
continues.
everyone on this watch does.
Yeah,
I
we
like
do.
long walks on the beach.
Has anyone ever been like, I hate long walks?
Yeah, no. I have a friend.
Actually,
I have a friend who
really?
doesn't like walking.
My brother hates sand,
I know.
Yeah, it does go everywhere.
so he doesn't like
Yeah.
going to the beach.
I prefer rocky coastlines myself.
Nah.
Far more character.
Yeah.
And you can like poke around and see
Yeah,
lots
fun
of,
little
lots of
tide
things
pools and things.
tide pools and
Yeah.
algae gardens and things like that.
Yeah.
Barnacle bites, though. No joke.
And Gabby, would you mind telling us a little bit about yourself?
Sure thing. Aloha. I'm Gabby Espino from Kailua, Hawaii, and I'm the navigator on board, and we're not diving, we are seafloor mapping, and that's my other job. And yeah, this is a great watch team. Happy to be a part of it.
Yeah, me too.
Yeah.
Happy to have you, Gabby. Yeah, Gabby is she's a twofer. She's doing all the things on board EV Nautilus.
I'm a bit jealous of you guys being able to sleep. So
We couldn't do it without you, though. We appreciate all your hard work.
Yeah, you have
course.
nav watches as well, right?
I haven't stopped any of the watches,
Yeah,
but yeah.
hard worker.
That's what we got to do. None of this area up here is mapped. We know so little about it.
Yeah.
So we're very fortunate to come out here and do this. And to give back to the Marshall Islands themselves so they know what the heck is out here.
Exactly, yeah.
Yeah.
It's so cool to have this experience, to be in these waters that have so little exploration before. Many of them have never been explored before, so we are the first people to get to see them. And as Gabby said, we are going to be using this data to help with management decisions in the Marshall Islands, which is really cool. I hope we see a shark
Niskins
as
at
we
50.
head north.
Okay.
Or head up.
Don't let me forget. Oh, it'll be your, you can do it.
What?
2 Niskins at 50.
Yeah.
Just
You
had a
won't forget.
question
I will.
I'll forget.
Okay.
if we're going to be doing any other dives on this expedition, and the answer is yes, we will. So this expedition, if everything goes to schedule, we should be back in Majaro in port on August 17th, and we have lots of dives planned between now and then. So hoping to get to explore some really other unique structures, some really other unique seamounts and ridges, and again, just learn more about these areas that have never been explored before.
Curious about, there's been some drawing on the whiteboard.
Yeah, I was wondering about that too.
They saw 16 halosaurs, which sounds about right.
Yeah, we definitely saw more than that.
2 cutthroat eels, and I don't know what this means.
What is that? Is
like
it names?
It's...
Is that initials for names, maybe?
Is Keone on the watch with... No, those are different watches.
Kelsey, Keone, Fanny, Brittany,
Feel like-
Giselle.
But these are...
Okay, yeah, we got to figure this out.
And then Hayden, yeah, okay. And
Brittany,
then
Fanny, Keone, Hayden.
Kelsey, Mike, Danny,
Brittany. No, I said Brittany, didn't I?
who's on nav for 4 to 8?
Isabella?
Isabella.
Yeah, Isabella.
Oh, so maybe Bella?
Okay, guys, in the lounge, you gotta fill us in what these marks on the whiteboard are.
And then they each have like points
Yeah.
of some sort, and whatever it is, it looks like Hayden's winning
Hayden's
with 25.
winning. Looks like Brittany is in second place.
And
Looks like
Danny has
is
no points.
No points. What's going on, Danny? We got to get in on this game.
Yeah, I'm really curious what this was.
Right, your screen is
I kind of like it.
acting up.
I think it should be like that all the time. I could try to fix it.
Oh yeah, it's the matrix down there for sure.
Yeah, that's kind of a cool vibe. Screen keeps flickering from normal to like a green wash
Yeah.
for those that can't see them.
Yeah, it looks like if you've seen the movies on the Matrix, it looks like the little green screen with the numbers, the zeros and ones.
What does it mean?
Oh!
Midwater fish.
Just had a comment saying they were all guessing how many fish they would see.
So then, okay, in that case, 16 plus 2 is 18, so Brittany won.
Brittany is
'Cause
our
she got it
winner.
right on the money.
Brittany Munson, big winner. Bow, 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 bow.
Does that mean Danny guessed 0 fish?
Well, it's not a 0, it's just blank.
It's just blank.
Oh.
So maybe Danny was like,
Not playing.
I'm not into this.
What is that?
Shrimp. Little shrimp.
Shrimp.
Oh, shrimp? Okay. Well, we already saw our fish before we got to guess, but how many fish do you think we saw? We saw way more than 18. I mean, I bet we saw
I might
40,
put it around,
50?
I don't know, that seems like a lot. I might put it around 20 or so, but I don't know
Okay.
for sure. I could go back and count, but I don't want to do that.
Yeah, I feel like at some points we would look on the screen and there would be like 5 or 6 halosaurs.
Well, that's a good
at
point.
any given point.
You could actually filter by halosaur, and you can count up quickly how many.
I feel like the filter never works for me.
Something we could do as we pass through this blue water.
Like I press enter and then it's like no events found.
Huh.
Let's see if I can figure this out maybe. I don't know.
Can I do this?
Yeah, you might be able to go to review.
Yeah, there we
Yep.
go. Okay.
Try that.
Leela is on it.
Yeah, siphonophore, right? Am I
Yeah,
correct?
that's a big one.
Okay.
Oh, cool.
They give me a heart attack.
Yeah, we are in kind of the lower deep scattering layer, the lower boundary of the deep scattering layer around here at 750.
Yeah, well, right now we're 631 meters below the surface.
Okay, so I only have 11 specific observations of halosaurs, but that doesn't necessarily mean we
If you put a
saw
comma,
11.
if you put a comma, can you also add fish? Like if you do comma separated, does it return both?
Yeah, we definitely saw more than that. I mean, just
No.
on their watch, they've got what, 16?
We had so many tripod fish.
Oh yeah, lots of tripods.
Yes, I don't know.
And sometimes when I'm putting a comment in and we saw a halosaur, like there might have been multiple in the shot. Saw a couple of those really cute batfish with the adorable little nose-looking structures.
Oh, yeah, we had like 2 of those, I feel like.
Yeah. I think we probably passed over a few more as we were looking for more important things, or more things that meet our objectives more.
Passing 600.
All of these fish are important.
Water chunks, right? Oh, look what they've been running.
Again, we have people, oh, little fishy. People from all around the world watching. Hello world. US, Canada, Martinique, Marshall Islands, woo woo woo. Guam, the UK, Germany, and Australia.
Something random, but it's been cool to have Val on board, is discussing, a lot of like earthquakes going on right now around the Pacific Rim. So I know we had that big one up in off of Russia. There's been some more active ones around like Indonesia, Chile.
Wow.
And just yesterday there was one off of Guam.
Oh, really?
Yeah,
I did not know that. Wow. And that one that was off of Russia was like the biggest one since the big one in Japan, right?
Yeah.
Wow. Something's happening, planet Earth.
Or in Russia. Yeah. We actually went over the little tsunami wave from...
We did?
Yeah, because it went all through the Pacific.
Huh.
So we were probably just like, little bump.
Yeah, little bump. Yeah, I didn't realize until that happened that like we're in the safest spot out here in the very deep ocean.
Yeah. That's weird to think about.
Yeah.
Then it'll just become water world.
Great movie. Who's that? Kevin Costner?
We go out to sea. Yeah.
Yeah, we are moving pretty fast. Already right around 550 meters. Heading up to the surface after a very interesting, successful dive on a ridge off of Bokak Atoll in the waters of the Republic of the Marshall Islands.
It's fine. It's all those rocks. Yeah. Oh, nice. Nice.
Oh, we just got a comment that there were 2 teeny earthquakes in New York, New Jersey a few days ago.
Yeah, they discovered a new fault line.
Oh, really? Wow.
Yeah, off Jersey. I can't do that accent.
Yeah.
Joyzy.
Joyzy.
There's a new fault line?
Yeah. They just discovered a fault line. Those ones are pretty old.
I thought I was safe in the northeast of earthquakes.
Well, the only earthquake I've ever actually felt was in Maine.
Really?
Yeah.
I don't think I've ever
I don't
felt
spend
one.
a lot of time in active fault zones.
We had one in Michigan once. It was a pretty small one, but I was like lying on my stomach on top of a baseball dugout.
Oh, wow.
So then I was like, did the dugout just shake?
Yeah.
We had one in Seattle probably 5 months
that's
ago.
Right. Yeah. And then another one my freshman year, so, you know, a few years before that.
Mount Rainier has been moving and shaking.
Yeah.
Yeah,
really?
a lot right now.
Let's not manifest anything.
It's Mount Rainier
It's
volcanic.
going to be the next month.
Yep.
Yeah.
Just remember, there's nothing you can do about it.
Yeah.
That is actually normally my like, what am I going to do?
Exactly.
You guys ever seen that movie Volcano with Pierce Brosnan?
Yes.
Oh, yeah.
It's one of my favorites.
Those movies always came out in twos. Dante's Peak
that was
right
Dante's
after that.
Peak.
Yeah, that was Dante's Peak. Volcano was the one with
yeah, Dante's Peak. That's
in
what I
like
meant.
LA, maybe.
Oh yeah, in the LA one they had to put the lava into like the drainage system or something and put out to sea.
That was actually funny.
What's that one called?
Dante's Peak was great. I watched that probably in the past few months. I love that movie.
Yeah.
Except the sad part with the grandma.
I bring that up every time and I'm like, what was she doing?
Yeah.
What was the plan?
Grandma
walking into the lava.
Yeah, trying to save the rest of them.
Yeah.
And they could have all made it out safely if they just listened to Pierce Brosnan early on. Just
As
listen
usual.
to the geologists, guys. They know what they're talking about.
Those disaster movies were a big thing, though. They had two meteorite movies, right?
Oh yeah, the Deep Impact with little Frodo.
Yeah, little Frodo and Morgan Freeman. Yeah.
I mean, I thought Twister was a documentary for so many years. It's so real.
I love Twister. It's way better than the new one, Twister.
Yeah, Twister's
I haven't seen the new one yet.
Yeah.
No spoilers.
Yeah. I'll just tell you the original's better.
Okay.
As most times is the case.
The new Twisters had my apartment complex in it when I lived in Oklahoma City.
No way.
That went up in the tornado movie.
Oh my God, that's so cool.
Yeah. The thing about Oklahoma is that they don't have any shelters in the city.
Oh wow.
So I've been in a few tornadoes there and they're just like, sorry.
Yeah,
nowhere to
that's
go.
because there's so many. It's Tornado Alley.
Yeah.
We get a lot in Northwest Arkansas as well. We had one not too long ago. It was the first time that I was like actually really scared. Like I got my dog and cat and we went into like the most inner part of my house and we took the mattresses off the bed and like sheltered ourselves. It was a little terrifying.
Yeah.
I've been filming more things in Victoria and I hate it because it's like I'm watching something and I'm like, oh, I know that place.
There.
Just like lots of I think there was like a few Netflix things that were filmed there. They're just using it like it's not necessarily set in Victoria.
Oh.
It's just like
just
I
because
can see
it's
them filming
pretty.
like Hallmark movies up there.
there
Oh my gosh.
Yeah.
The new Batman. I mean, it's not that new anymore. It was from like 2022. But they shot that in Chicago because
Cool.
movies will actually use Chicago for Gotham a lot. And I can never decide if I'm offended by that or not. It's like, this is the worst city in the world. It's dark and dank. Let's shoot it in Chicago. But they did a really good job and it looked really nice.
Yeah.
So.
We don't get too many in Northwest Arkansas, although they did, I think, did some scenes from Ozark, the series.
Yeah. The big one in Chicago recently is The Bear, which everyone from Chicago is deeply obsessed with, myself included.
It's a great show.
It's so good.
Yeah, Hawaii has a lot of movies filmed there.
I'll bet.
Jurassic Park, right?
Oh yeah, just that's like the first OG movie.
Yeah.
Gilligan's Island.
Yeah, that was on
We
Coconut
were just
Island.
talking about.
Yeah, I just learned about that.
Yeah. When I was in high school, they were filming Lost. And I just became accustomed to seeing all those actors in my town.
They filmed which one was it? One of the True Detective HBO series in Fayetteville, and they filmed it in my neighborhood, and so me and my roommate would like go walk the dogs and like accidentally walk through a live scene.
Oh no.
Oh no, we're on camera. Oh no.
Well, were you in an episode?
No, but they did send out a call for extras, and several of my friends were extras in it.
Nice.
About to pass 400 meters.
Copy.
You would see like all the actors and stuff out in town at restaurants and bars and things like that. It was pretty neat.
Got a few bird visitors on the deck this morning.
There was one in the hangar. I just threw him out of the hangar.
Really?
What?
Yeah.
See?
I closed
Which
the hangar.
kind?
The
Oh,
black
they're
one.
getting.
Oh,
I
the little
closed the
shoe one?
hangar, and I was like, oh, someone else the hangar
They're
open
getting
last time. Close it,
brave.
and I look over, and there's a bird in there just looking at me. I was like, you're not supposed to be in here. So
There's
I opened
never
the door
just one.
and like shoot it out,
Yeah.
and then close the door.
You'll be going to do your laundry next, and they'll just be in the dryer.
Just staring at you.
Yeah. I walked right by it, didn't notice it, and then closed the door and looked back, and I was sitting on the rail. I was like, wait, you've been there this whole time?
There was one right when Mariana and I were coming out for shift.
Yeah.
I think it was a booby that was landing on the one of the cranes.
Oh wow. Yeah.
It was a little.
Yeah, it's probably easier for them to land when we're not moving, when we're stationary or somewhat stationary in these dives.
Yeah, he's just sitting there.
Oh.
Yeah.
Whoa.
I had to like hit him with a broom to get him, or like gently touch him, with a broom to get him to go. He looked a little bit
Get outta here.
disoriented. It's like he landed and got seasick. Uh, 17, yeah.
Oh, I think that was deck. Sounded like Drew.
Is that coming through my headphones, or is it
No,
over
it's
there?
something out here. I don't know
Okay.
where it's coming through. Fingers crossed for some sharks as we ascend.
Mm.
Bum bum. Bum 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 bum.
I don't know.
Do you guys have a favorite shark?
Oh man, mine is, that's tough. Either the whale shark or hammerhead, great hammerhead.
Good choices.
Hammerheads are cool.
Although I also really love nurse sharks. They're like little puppies of the sea. They're
I
adorable.
know, they're so sweet.
I got to do this teacher fellowship in Bimini, Bahamas, and we got to just interact with sharks in their native habitat. We went to this little secret spot in the mangroves. It's a little nursery for nurse sharks and lemon sharks, and there were probably, I don't know, 50 to 75 of them, little babies, and they were just swimming all around you. They were swimming through your legs and bumping into you, and it was just amazing.
So sweet.
Yeah. Sharks get a bad rap, but they're very, very important for marine ecosystems. And we have unfortunately decimated their populations. What about you, Leela? Favorite shark?
Making what?
I was going to say, did you know that sharks can wiggle up onto the sand and back out to sea?
Ooh.
I did not.
Ooh.
Yeah, when my dad was little, he was walking on the beach, and there was a little dog on the beach too, and a shark came up onto the sand, got the dog,
Oh my
no.
God.
and wiggled back out.
It's like those orcas that can go up on the beach to
Yeah.
get
Yeah.
Get
Um,
seals and things, yeah.
I love thresher sharks.
Threshers
Yes.
with a big whip-like tail.
and they
Yeah,
Yeah.
they're so cool.
But I also really do love whale sharks.
yeah.
I saw them at the aquarium in Japan, and they were just
Oh
incredible.
yeah, they're incredible. If you ever get a chance to snorkel or dive with them, it is out of this world. They're so huge and massive, you just really can't.
I actually did when I was a little kid, but I got too freaked out and had to go back on the boat.
Yeah, it's scary.
I was like in the water and I looked down and there was a shark and I was like, nope, goodbye. Which regret now, but yeah, you know. What about you, Mariana? You got a favorite shark?
I'm gonna say the hammerhead.
Yeah.
Solid.
Yeah, they're really interesting. Such cool adaptations.
Go for nav.
Front row, favorite sharks? Start with you, Dan. Favorite shark?
Ooh, I mean, everyone already picked hammerhead. What are the really deep sea ones? Is that the Greenland?
Greenland?
Greenlands?
Yeah,
because they're just so ancient.
Yeah.
Those are the ancient ones.
They're so cool.
The eyes are a little scary, but yeah.
Yeah, they're kind of cloudy.
They have like parasites on them too. Right?
Yeah.
Yeah. I don't even use them. Oh, maybe.
Don't forget the most famous shark of all.
Right.
Baby shark.
Oh my God.
Not what I thought you were going to say, but I like it.
If you live through the pandemic with children, you know what I'm talking about.
Yep. What about the, what was the one on with Katy Perry, like Left Shark? Do you guys remember that?
Yeah. Awkward left shark or whatever.
Did you guys know Greenland sharks have been found with the remains of moose, horses, and reindeer in their stomachs?
What?
Yeah.
I guess, did they just like drown and then the sharks eat them?
Well, okay, admittedly I'm on Wikipedia right now, so take this all with a grain of salt.
Yes, students, that is not a reliable source.
Not a
for your
reliable
papers.
source. It is a good place to find primary sources, which then are reliable.
Exactly.
But don't use it as a source in and of itself. But their citations are the Florida Museum of Natural History, which is solid. But then there's a second, the sentence after that says polar bear remains have also been recovered, though these are thought to be of bears that died from other causes.
Okay.
Which to me implies that they think that the moose and such
May
did
have-
not die from other
Right,
causes,
yeah.
died from the cause of Greenland shark.
So maybe the Greenland sharks are like scavengers a little bit?
Yeah, I think so. Anyways.
Well, moose go swimming, right? Like they,
sounds like they
like sometimes they're found swimming between islands.
they
and
definitely
stuff.
swim.
That's why orcas are predators of them.
Yeah.
But
Really?
I want to,
I didn't know that.
Yeah.
That's wild.
That would be wild to see moose
Right,
taken out by orcas.
Be a hungry orca.
But I want to repeat to take this all with a grain of salt. I am an intern and not a shark expert.
Yeah, the transient orcas are pretty cool.
Orcas are pretty cool.
I love the orcas.
They are. They're amazing, but also quite scary.
Do you know that they can eat the livers out of great white sharks?
Yes,
They do like a surgical strike.
Yes, they do it on purpose, and they'll like leave the rest of
They
it.
sure do.
Because it has more nutrients, I assume, or?
Yeah, it's full of all the good stuff.
It's really oily, which is what they use to control buoyancy in the water.
Someone just said salmon sharks. Have you guys seen those? They look like what a great white shark pet would look like. Like if you domesticated a great white shark, they're like little miniature ones. They're so adorable. Definitely give it a Google if you haven't seen them. They're so cute.
Where do they come from? I've never heard of those.
Northern Pacific Ocean.
Looks like baby shark.
It is. It's baby shark.
We found it. The baby shark.
Huh. Can't wait until we can talk whale using AI. Aren't they trying to do that now?
That's crazy.
Give us your best whale. Great job, everyone. If we keep doing this, we'll summon a whale.
Yeah.
Probably humpbacks first.
Escape.
I did see that. Can't remember where it was published, but I saw something about the whales have been singing less because they're having to devote more energy to searching for food because there's less food resources available due to global climate change.
And ship noise can be inhibitory as well.
This is sad.
The Natural History Museum recently acquired a megamouth shark.
Whoa. Oh,
that's the head.
Wow.
It's too big to actually fit, but
that's cool.
All of the large format collections are stored like this.
Whoa.
They call it the morgue.
Yeah, I can see why.
Yeah.
Whoa. What is that?
That's a goosefish. Or
that
a
is
monkfish,
like something out of a nightmare.
Yeah, these are North Atlantic.
Okay.
Passing 200.
I will be in DC like the first weekend of February, so I'm going to request a behind-the-scenes tour, Steve.
I'll be there.
Awesome. Let's see, where are we? Oh, we're getting very close. 190 meters, 92 meters below the surface. So again, those of you just tuning in, we are finishing up our dive just off a ridge off Bokok Atoll in the newly founded National Marine Sanctuary of Bikar and Bokok in the Republic of the Marshall Islands. This is Expedition NA174, and we have many more dives to come. So although we are finishing this dive, please continue to tune in to nautiluslive.org and follow along on social media where you can see some amazing updates from these dives. All right, we did favorite shark. What about favorite whale? Everyone, is it the orca, I would assume, for a lot of people?
Orcas are dolphins.
Sperm whales.
True. Oh, yeah, they're pretty amazing looking. Those look like the ones from like Moby Dick, right?
Yeah, and they have like, they're the only ones with teeth, and then they dive and hunt giant and colossal squid.
Super cool.
Awesome.
I really want to see a bowhead whale in my life.
Bowhead, I don't
like
know that one. I want to look that one up.
Pointy thing?
It's like
I would love to see a blue whale.
baleen.
That's like
Mine are
life goal.
Orcas, but I think humpbacks are a close second.
Yeah.
Rye, do you get northern residents
wow,
around
those
Victoria?
bowheads are wild
Yep.
looking.
So cool.
My dad used to work as a whale watching guide when I was a kid, so
Saw a
we
lot.
had a binder with all the names and all the pods.
Okay,
JK
yeah. Yet another reason I'm coming to visit you, Rye. We're going to go whale watching when I come visit you.
Yeah, I haven't been in a very long time.
When I was at my program right before this, which was on San Juan Island, which is an island in the Salish Sea, kind of in between Canada and the US. It was the last evening, and we were all sitting on a rock by the water and just like chatting. And then like 15 feet away from us, a gray whale pops up and then goes back down. And there was a lot of screaming.
I bet, yeah.
I had humpbacks, like we were just sitting on a little 14-foot skiff, and two humpbacks came up beside us, like we didn't know.
Whoa.
Amazing. And then they came up beside us, and it was so close you could see like the barnacles growing on them.
Yeah.
Whoa. And
so
it was
cool.
so loud there.
I got to see something similar in the Sea of Cortez, just off Baja, Mexico, some gray whales. We were in like a very small boat, and they just like were right there. I could have reached out and touched it if I wanted to.
Awesome.
Also, I really love belugas and narwhals. Narwhals, I didn't think